All right, well, thank you so much for having me. My name is Tanya Cheeke. I'm an assistant professor of soil microbial ecology at Washington State University, and my lab is located on the Tri-Cities campus. The research I'm going to be presenting today was some of the graduate student work done by Casey Sifazari, who was a master's student who recently graduated, and the current work is being done by Madeline Luke, who's also a master's student in my lab. And none of this work could be done without the many undergraduates who've helped at WSU Tri-Cities from the viticulture and enology program, as well as through the biology department. Cooperators on this work are Dr. Michelle Moyer, a WSU viticulture extension specialist who's been helping a lot with this project, and then Sarah Del Moro and Catherine Jones from Bleo Co-op. All right, so I wanted to give a little bit of background on myself. So I'm actually from the Pacific Northwest. I did my undergraduate degree in sustainable agriculture and environmental science at the Evergreen State College. And I did my PhD at Portland State University, studying the effects of different genotypes of corn on mycorrhizal fungi in the soil ecosystem. So here is a picture of one of my field studies. I did postdoctoral work in Sweden and in Indiana, where I looked at the role of mycorrhizal fungi in soil biogeochemical processes and also in ecological restoration. And since I moved to the Tri-Cities about 4 years ago, I've expanded some of the research into viticulture. And so this is a new system for me, but it's been really fun to learn more about it. So in my lab, we study the impact of soil microbes on plant growth and soil processes, and we use a combination of molecular tools and microscopic analyses to do our assessments. And one of the things that I've been focusing on since I got to WSU is whether or not inoculations with some of these soil microbes can be helpful or beneficial in sustainable agroecosystems, and also for getting native plants established in disturbed ecosystems for ecological restoration. So the soil microbiome is made up of a complex group of soil organisms that are important for both soil and plant health. In the soil ecosystem, you have this really diverse community of organisms. We have symbiotic soil organisms like arbuscular mycorrhizal fungi, ectomycorrhizal fungi, and nitrogen-fixing bacteria, which form symbiotic relationships with plant roots and can help improve nutrient uptake. There are decomposers, like if you ever go hiking and you see some little mushrooms growing in the woods, some of those are decomposers, and so they break down organic material and recycle nutrients. There are endophytes and pathogens that live on and in the plants that can have a positive or negative impact on plant growth. And then there are larger organisms like microarthropods, nematodes, and protozoa that are also important for nutrient cycling. And all of these different organisms can be impacted by a variety of factors, including management practices. So some of these organisms are especially sensitive to tillage, for example, that breaks up soil. They are also affected by the type of plant that's grown and the diversity of plants that are grown. So here, a monoculture versus a diverse plant community. And at different times in my lab, we've studied pretty much all of these different organisms, but today I'm going to be focusing on the arbuscular mycorrhizal fungi. So arbuscular mycorrhizal fungi, or AM fungi, form symbiotic associations with plant roots, and they're found in almost every plant that you'll find out in the environment. They're actually thought to be the most ancient symbiosis and are thought to have helped plants establish on land over 450 million years ago. So this is a very common symbiosis. And so in the symbiosis, the mycorrhizal fungi are in the soil, and they form symbiotic relationships with plant roots. The plants are photosynthesizing and give carbon down into the roots that the fungi use. And then the fungi act as an extension of the root system. And so these little gray threads here are little cartoons of the fungal hyphae, and they go out into the soil beyond the root zone and can collect nutrients and water for the plant. And the arbuscular mycorrhizal fungi are especially known for helping improve phosphorus acquisition and uptake. So the mycorrhizal hyphae in the soil really expand the surface area of the plant roots. And here's what they look like under the microscope. And then they have these little structures here, which are the site of nutrient exchange. So by taking root samples, we can look at how colonized the plants are, for example. And one of the things that's interesting about this symbiosis is that the mycorrhizal fungi are obligate symbionts, which means that they require a host for their growth. But they can have varied impacts on plant growth. In some situations, they can improve plant growth, but in other situations, they can actually reduce plant growth. And in still other situations, there can be neutral impacts. So the benefits of using mycorrhizal fungi really depend on a lot of different factors, from soil type to soil nutrient availability, even to the genotype of the plant species and the fungal species, and where the fungi came from. So there are a lot of different aspects to this. We also know that common management practices can disturb naturally occurring fungal communities in the soil. So tillage is one of the ways that the mycorrhizal fungi can be negatively impacted, because in part because of those thin hyphal threads that they have in the soil. Those can get chopped up through tilling. Fungicides can also have a negative impact on mycorrhizal fungi because they're antifungals. And the site history can have an impact on the mycorrhizal communities. How often it's been tilled, what the previous crop was, and so on. Here's some data from a study that really shows how management practices can reduce mycorrhizal spore density and species richness in the soil. So here on the y-axis is the number of spores per gram of soil in grasslands, vineyards, and cornfields. And then these different colors just represent sampling depths. And what they found was that overall in the cornfields, there were much lower levels of mycorrhizal fungi and lower species richness than in the grasslands and in vineyards, for example. And so one of the things that people have been more and more interested in, especially when they're thinking about biofertilizers or incorporating aspects of sustainable agriculture in their system, is using some of these mycorrhizal inoculant products to reintroduce fungi into soil where the mycorrhizal communities have been disrupted. The nutrient availability in the soil can also impact the plant-fungal symbiosis. So in fertilized soils, the mycorrhizal fungi don't tend to be as beneficial, because in the symbiosis, the plant is giving carbon to the fungi. So they're investing in the fungi to make this network in the soil. And then the fungi in turn help improve nutrient uptake. If there are already a lot of nutrients freely available in the soil, the plant oftentimes has less investment in that fungi, or doesn't get as much benefit from the fungi. So the mycorrhizal fungi tend to be more beneficial to plant growth in unfertilized or low fertility soils. And this is a study that was done by Paul Schreiner in 2007, where he looked at the impact of mycorrhizal inoculations in high and low phosphorus soils using Pinot Noir. And what he found was that there was an increase in shoot length and also in phosphorus uptake in Pinot Noir vines, but only in the low phosphorus soils. So this again shows that the benefits of mycorrhizal fungi can vary, and they tend to be highest in low nutrient soils. So when I got here, I moved here about 4 years ago. I met up with Michelle Moyer, and we were talking about how the type of research that I do might be able to be beneficial or useful for wine grape growers in the area. And so we sent out a survey and wanted to get some feedback on whether this would be interesting. And about 75% of the respondents said they would be interested in seeing WSU conduct research looking at the effects of these different types of products on wine grape growth in this area specifically. And this is because there are a lot of these different products on the market, and they contain all sorts of different microbial organisms, from nitrogen fixing bacteria to arbuscular mycorrhizal fungi. But oftentimes there's little consistency in results because these products vary quite a bit in what they contain, how many strains of organisms they contain, what systems they work best in. And sometimes they actually don't even work at all. I've been involved in a couple of projects where somebody's inoculated with mycorrhizal fungi, we harvest at the end of the experiment, and there's no colonization. So sometimes these products, it's really hard to know what to use and when and how. So the efficacy of these remains unclear. It's also unclear under what environmental conditions they might be most beneficial. And in the case of wine grapes, for which wine grape varieties or cultivars, are there certain cultivars that are more responsive to mycorrhizal fungi than to others? So what we did, we are working on 2 greenhouse studies, one that was led by Casey Stefazari, that is completed. And this was to determine whether inoculation with mycorrhizal fungi increases grapevine growth or tissue nutrient content when grown under different phosphorus conditions. And we used 2 different wine grape cultivars, a red, merlot, and a chardonnay. And then in an ongoing experiment that we're going to be doing this spring, we're evaluating the impact of different mycorrhizal inoculant products versus local mycorrhizal fungi on wine grape growth and tissue nutrient content. And we're doing this in 2 ways. One, we're going to follow the manufacturer's recommendations and add exactly how much they recommend. And then we're also going to standardize by mycorrhizal inoculation potential, which I'll talk about a little more later in the talk. And here's our research team. So I've been working with Dr. Michelle Moyer. This is Casey, who recently finished their master's at WSU. Madeline Luke is currently working on the project. And then a team of undergraduates from biology and the viticulture and enology program have been helping with this research. All right, so the first thing we wanted to know is, how does inoculation with mycorrhizal fungi growth of merlot and chardonnay under different soil phosphorus conditions? We were curious to know whether different wine grape cultivars respond similarly to the same inoculation treatment, or whether growth response to mycorrhizal fungi varies by cultivar. We also wanted to know whether inoculants with or without phosphorus fertilizer applications would impact plant growth and nutrient uptake, and whether there were benefits of incorporating both inoculants and phosphorus fertilizer, or is one generally better than the other. And the goal of this work is to really start to understand the role of mycorrhizal inoculants on grapevine growth and nutrient uptake to help inform management strategies aimed at incorporating biological products, because that's becoming more and more popular. People are wanting to use some of these products. A greenhouse experiment, and we had the 2 different cultivars, merlot and chardonnay, and we had 4 different treatments. And throughout the talk, the controls are going to be in pink. So these controls didn't have any mycorrhizal inocula or fertilizer added. The mycorrhizal fungal treatment is in blue, and this is only mycorrhizal fungi were added. The pea fertilizer treatment is in yellow, only phosphorus fertilizer was added. And then we have a combination of mycorrhizal fungi and phosphorus fertilizer in green. And we had 15 replicates, and so there were 120 pots in this experiment. We grew the plants for 5 months in the greenhouse, and then we measured the effects, or we measured plant growth every month. And then at the end, we harvested the plants and separated roots and shoots. And we also collected plant tissue for nutrient content and scored mycorrhizal colonization levels in roots. And our hypothesis was that inoculation with mycorrhizal fungi would improve growth and tissue nutrient content in both grape cultivars, but that the benefits of mycorrhizal inoculation will be highest in the no fertilizer treatment. To do this work, we chose to use a fungal inoculant product called MycoBloom, which has 7 different mycorrhizal fungal species. And we're using this product because I've used this one a lot in the past, and it's one of the ones I trust. I know that it tends to work to colonize the roots. It was developed at Indiana University by a group of mycorrhizal researchers, and these fungi were originally isolated from perennial plants. And they've been used in more than 15 years of peer-reviewed publications. So it's been shown to improve growth of lots of different types of perennial plants. And so I wanted to see if it could have benefits for wine grape growth in Washington. And the way that we test this is we inoculate the pots. So we'll add the mycorrhizal fungi to the pots, and then we'll put in a plant, and then we'll grow them. And then what we do is we measure how big they grow with inoculation versus without. So if plants grow larger when they're inoculated, we would say this has a positive growth response to mycorrhizal fungi. So here's an example of basil growing with mycorrhizal fungi than without. So this is an example of a positive growth response. It grows better with inoculation than without. There are also lots of plant species that are not responsive to mycorrhizal fungi. So even if they get colonized, there usually aren't any observed differences in growth. And to make things even more complicated, their different plant species can respond differently to the same mycorrhizal fungal species. So this is a study that was done using only one species of mycorrhizal fungi. And then anything above this 0 is a positive growth response, and anything below here is a negative growth response, meaning that these plant species grew worse when they were inoculated, and these plants grew better. And so different plant species can respond differently to the same mycorrhizal fungal species. The same plant species can also respond differently to different mycorrhizal species. Here's an example of Bromus inermis. It's a grass. And here what they're showing is all these different colors indicate single mycorrhizal species that they inoculated with. And this single plant grew worse sometimes when it was inoculated with certain species, and grew better other times when it was inoculated with certain species. So this just really highlights that a lot of these results can be context dependent. So it's important to understand whether the fungi that you're inoculating with are actually going to be beneficial for your plants under your particular conditions. Different cultivars of the same plant species can respond differently to the same fertilizer and inoculant treatment. This was some work that I did when I was doing my PhD, and this is 2 different corn cultivars, and they were grown in no fertilizer, low fertilizer, or high fertilizer treatments with different numbers of mycorrhizal spores added. And what you can see is even though they're grown all in the same condition, one cultivar has a lot higher levels of colonization than another cultivar, and that was consistent across the fertilizer treatments. So even within the same plant species, corn, mycorrhizal colonization levels can vary based on the genotype of the plant or the cultivar. So for our research questions, we wanted to test a common red and a common white wine grape to see how they respond to a mycorrhizal fungal inoculant, and how the addition of phosphorus fertilizer with and without mycorrhizal inocula might impact plant growth, mycorrhizal fungal colonization levels in roots, and tissue phosphorus uptake in each cultivar. So here's a picture of our greenhouse experiment. We had the 2 cultivars, Merlot and Chardonnay, 2 inocula treatments, pots were either inoculated or not, and 2 phosphorus treatments. They either got phosphorus fertilizer or not. And then we had 120 pots in the experiment, and we collected monthly growth measurements over 5 months. The way we set up the pots, we had these 4-liter pots, so they're fairly tall pots. And what we did was we filled, 90% of the pot was filled with an autoclaved sand soil mix. And so we collected local field soil, mixed it 1-to-1 with sand to improve drainage. And then that's the background material that's in all the pots. And then the pots that get the mycorrhizal inocula get a thin layer of mycorrhizal inocula placed at the root zone. And then we used a dormant non-rooted cutting of either Merlot or Chardonnay to put in there. And then we also did a microbial filtrate where we extracted the bacteria from the inocula so that we can account for non-mycorrhizal microbes. So the only thing that varies is the mycorrhizal fungi. We collected the background soil from the research station in Prosser. The field soil had an original Olsen P content of 31 parts per million, which was a little higher than what we were looking for the greenhouse experiment, because in high phosphorus soils, the mycorrhizal fungi tend not to be as beneficial. So what we did was we mixed it 1-to-1 with sand and autoclaves to kill the resident organisms. And then the final soil mixture was an Olsen P of 10 parts per million. And this, the phosphorus concentration in the background soil is within the range of soil phosphorus of Washington vineyards. So we talked with local experts and found that the range of soil P ranges from 8 to 40 parts per million, with 8 to 15 seeming to be representative of the average vineyard soil. And then here are some different soil P numbers from different areas around Washington. And then this was the Prosser number. So we got our calloused unrooted cuttings of Merlot and Chardonnay from Inland Desert Nursery. They provided those for our research. These are self-rooted vines. And so we planted them into each pot with the roots, and we got unrooted cuttings because we didn't want them already colonized with mycorrhizal fungi. We really wanted to look at how this fungal inoculant impacted growth of the Merlot and the Chardonnay. So they were planted, the root zone was in the mycorrhizal inocula, and then we added phosphorus fertilizer to the phosphorus treatments. And then we also had a low nutrient fertilizer that we added to all pots so they wouldn't be stressed in other nutrients. And we hypothesized that the mycorrhizal inoculant would increase plant growth and tissue phosphorus in both grape varieties. And that we thought that the benefits of inoculation would be highest in the no phosphorus fertilizer treatment. So what we're looking at is the difference in growth here. So we would expect to see a bigger benefit of adding mycorrhizal fungi compared to the control in the no fertilizer treatment. Whereas if you're already adding a bunch of phosphorus and then you also add mycorrhizal fungi, we wouldn't really expect to see a benefit. And then we also thought that the Chardonnay and Merlot would vary in their response to mycorrhizal inoculation. And this is just based on previous studies I've done with other plant species where they do tend to vary based on cultivar. Okay, so does mycorrhizal inoculation affect growth or foliar tissue P concentration of Merlot or Chardonnay? So I'll be presenting the data that we collected for this first experiment. We collected shoot length every month. At the end, we harvested the plants, and I'll be we did tissue nutrient content and mycorrhizal colonization of roots. Okay, so here to orient you on the y-axis, this is shoot length in centimeters, and then this is days after planting. So we started measuring 60 days after planting and then collected data each time period. Throughout the talk, the controls are in pink, the AMF inocula, the mycorrhizal inocula, is in blue, the mycorrhizal plus fertilizer is in green, and fertilizer only is in yellow. And we started seeing treatment effects in Merlot between 90 and 120 days. That's where we really started seeing the plants responding differently. And what we found in the Merlot was that the addition of the mycorrhizal inocula increased shoot length of Merlot, but only in the no fertilizer treatment. So these different letters here mean that they're significantly different. So adding mycorrhizal inocula led to significantly longer shoot lengths compared to the uninoculated control. But then in the phosphorus fertilizer section, we don't see a benefit, at least in shoot length, of adding mycorrhizal fungi. In fact, they grew just a little bit smaller. And in Chardonnay, on the other hand, we found no effect of mycorrhizal inoculation on shoot length. So here what we see is at the end of the experiment, the plants were all about the same. The plants that got phosphorus were a little bit bigger, but there was really no difference. Adding inocula didn't seem to have any effect. Okay, then we looked at shoot biomass. So this is shoot biomass at the end of the experiment. So the plants were harvested and dried and then weighed. And here again, we see that the mycorrhizal inocula increased final shoot biomass of the Merlot, but only in the no fertilizer treatment. So here we have the control, and then this different letter here indicates that these are significantly bigger or more biomass for the shoots. And then we don't see a difference here when phosphorus fertilizer is added. And similarly to shoot length, we don't see any effect of mycorrhizal inoculation on Chardonnay. So they grew just as well with or without mycorrhizal fungi in the no fertilizer, and with or without mycorrhizal fungi in the high fertilizer treatment. One of the things that we thought was most interesting is that when we looked at root biomass, we actually found Merlot root biomass was highest in the combination of mycorrhizal fungi plus phosphorus fertilizer treatment. So there was no difference in root biomass in the control or the mycorrhizal treatment or the phosphorus fertilizer treatment. But when you added mycorrhizal and phosphorus fertilizer treatment together, we saw a bigger root biomass. And so this could be important because it could help improve nutrient uptake in the soil if you have a bigger root biomass. And it could also maybe be helpful for improving vine establishment if you're interested in having a bigger root system. And when we saw this result, and I'll talk about this a little bit later, this has also been seen in other studies where a combination of mycorrhizal inoculant plus commercial fertilizer can lead to higher root biomass in several different plant species. So I thought that was pretty interesting. And then in Chardonnay, we again didn't see any effect of the mycorrhizal inocula on root biomass. Then we looked at tissue phosphorus concentration. And so what we're seeing here is this is phosphorus concentration of the above ground plant tissue. And in the control, in the mycorrhizal inoculated, and then here is where the fertilizer was added. And so this is fertilizer only and mycorrhizal fungi plus the fertilizer. And so in the overall statistical model, we had effects of phosphorus and mycorrhizal fungi. But when you look at these pairwise comparisons, there's not statistically different here, but the phosphorus concentration of the mycorrhizal fungi is a little bit higher, and it increased tissue phosphorus from 0.1 to 0.18 parts per million. And so sometimes these results might not be significantly different, but could be functionally important. And so if this is something that you're interested in, and if you're growing in a low nutrient soil, mycorrhizal fungi, it does appear to be able to help increase phosphorus tissue content. But in the fertilizer treatment, we didn't really see that. We saw just a slight increase. And then in the Chardonnay, we also didn't see any significant the low fertilizer treatment, the mycorrhizal inocula did help increase just a little bit. And no effect in the high fertilizer treatment, in the phosphorus fertilizer treatment. All right, and then finally, we looked at the mycorrhizal colonization levels in roots. And so these plants were grown for 5 months in a greenhouse with the mycorrhizal inocula. The control and the pots that only had phosphorus fertilizer didn't have any colonization. So that was good. That means our background soil was sterile. And then in both varieties, or in both cultivars, we had really high levels of mycorrhizal colonization in the inoculated pots. So this was 75 to 85% colonized. Whereas when phosphorus fertilizer was added, that colonization was reduced significantly. So it went from more than 75% colonized to less than 20% colonized just by adding the phosphorus fertilizer. And so even in situations where you might not see strong effects of mycorrhizal fungi on plant growth, and again, this was a short-term greenhouse experiment with young vines. So with perennial plants, you can see effects of mycorrhizal fungi and other soil organisms can vary over time. But what this shows is that adding phosphorus fertilizer can significantly reduce mycorrhizal colonization levels in roots. And mycorrhizal fungi aren't just important for phosphorus uptake, for example. They can help improve drought tolerance, increase access to micronutrients, and so on. So this could be interesting if you are looking to increase mycorrhizal colonization levels in roots. You want to be careful about how fertilizer impacts that. Okay, so for this first experiment, I'll go over the summary of results here. So for merlot, we saw that adding mycorrhizal inocula increased shoot biomass and shoot length, but not in Chardonnay. We also found in merlot that adding mycorrhizal fungi to the phosphorus fertilizer treatment led to larger root biomass, but no effect of mycorrhizal inocula on Chardonnay. And then when we looked at tissue phosphorus content, there was a little bit of an increase with mycorrhizal fungi in the no fertilizer treatment, but we didn't see that in the Chardonnay. And then for both cultivars, adding phosphorus fertilizer reduced colonization by quite a bit. So what we found was that results vary by cultivar, and we only had 2 cultivars in the study, but I think it would be interesting to try it with a lot of different varieties and also on different rootstocks. So what we found was that merlot had a stronger growth response overall to mycorrhizal inoculation with this particular product than Chardonnay. And despite similar levels of mycorrhizal colonization. So they both got similar levels of mycorrhizal colonization, but only merlot had a stronger growth response. So in thinking about what this might mean, and this is just thinking about it, it could be that at least at this point in time, Chardonnay has lower phosphorus requirements because there's not that much of a difference between the phosphorus treatment and the control. So they seem to grow pretty well in all treatments. Whereas in Merlot, when you added the phosphorus, they grew a lot bigger. So indicating that they're not getting enough phosphorus maybe in the control. So that's just one idea of maybe why we might have seen those differences. And these cultivar specific results are consistent with other studies with other plant species, and results can also vary by rootstock. So there are more and more studies coming out looking at how different rootstocks respond to mycorrhizal fungi. So in the last part of the talk, I want to talk about the second experiment to test whether plant response also varies by mycorrhizal product. So in this study, we had a highly controlled experiment with only one mycorrhizal product, but there are lots of different mycorrhizal products on the market. So what we did, or what we're doing this spring, is we are evaluating the impact of different inoculant products on wine grape growth compared to the local mycorrhizal fungi that are already here in the soil. So here we have 4 different products that you can buy off of the internet or in stores, and I'll talk about those in a minute, compared to the fungi from local remnant soil. So we went to a local undisturbed natural area and collected soil from underneath sagebrush because that's a long-lived local perennial. We also went out to a local vineyard, an established vineyard from a row that had, or from an area that was for 50 years. So this is an established vineyard that we collected soil from. And then we are looking at just background mycorrhizal colonization levels in field soil. So just local field soil where you might establish a vineyard eventually. And in this experiment, we're focusing only on Merlot to see how they respond to all these different treatments. So in this experiment, we're going to have almost 400 pots in total, and that's going to be set up this spring. And we hypothesize that plants will vary in their growth response to these different products. So we've done some preliminary research on this. We've collected the live background soil from near the WSU campus vineyard. And so we just collected soil from campus, and that could be potentially representative of future vineyard soil. So if you were going to establish a vineyard in that soil, what are the mycorrhizal communities like already? Then we picked a selection of inoculant products that are readily available to growers through local stores or online. And then we're curious to see how these inoculant products interact with resident soil organisms. So in the last experiment, we sterilized the background soil and really focused on differences in plant growth between these 2 cultivars. Here, what we're doing is testing when you add these inoculants to existing fungal communities in soil, do they do anything? Are they beneficial? Can they even establish? And so what we're going to be doing is testing whether inoculants increase mycorrhizal colonization levels compared to background soil, improve plant growth, or foliar nutrients compared to the resident organisms. And then how does this commercial inocula compare to inoculation with local soil inocula? So one of the things that people are doing in the restoration world is they're taking soil from an intact remnant community and moving that soil into a disturbed area to help some of those plants get established. And so this is a similar system, a similar approach of basically a soil microbiome transplant, where you can take soil and its existing organisms from either a remnant area or an established vineyard, and then see if that helps in a new vineyard. So we're gonna test that here. So these different mycorrhizal products varied in the number of fungal species. So here are all the different products that we're testing, and also local soils. And they vary from 1 fungal species up to 9 fungal species. They vary in propagule density. And so propagules is just like spores, things that will germinate and associate with plants. So they vary from 25 spores per gram up to 300 spores per gram. And so we were curious, based on all of this variation, how will this affect mycorrhizal colonization levels in roots? And so what we've done is we've done a bioassay. It's called a mycorrhizal inoculation potential assay And what we might expect is that this product that has the lowest number of spores maybe would have the lowest colonization, and then this product that has the highest number of spores maybe would have the highest colonization. So we're gonna check that. And one of the reasons we're doing this is because this number doesn't mean that these spores are viable. This means that at some point somebody measured the number of spores per gram, but not all of them are viable. And so these products could be wildly different in terms of colonization, or they could be the same. So we'll see. Okay, so we conducted a mycorrhizal inoculation potential assay earlier this spring. And this is a really common method in mycorrhizal ecology work. So you just grow corn plants. They're a nice biotrophic plant. You grow them with all the different inocula for 30 days. So they're all growing with the same plant in the same soil with the same amount of inocula. And then you quantify mycorrhizal colonization levels and roots. So every one of these pots got 1 tablespoon of a particular product, and then we had replicates. And then we clear and stain the roots, and we look at the fungi under the microscope and stain them so we can quantify them. All right, and here are the results. So with these different treatments, here we have percent mycorrhizal colonization of roots. And what we found was there was a lot of variation in colonization. So remember, all the pots got the exact same amount of the same volume of inocula, but because the propagules vary so much, you can get some, and the species, you can get variation in results. So here what we found was the mycobloom had the highest level of potential, and the local field soil had the lowest. And so if you remember, we had, I just mentioned this mycobloom had the lowest spore density, and this mycogold had the highest spore density. But what we found was that despite this variation in propagule density, mycobloom had the lowest spore density, but the highest levels of colonization. So what this shows is that the propagule density on the bag doesn't necessarily correlate to higher levels of colonization. So this product worked just as well as this product in terms of mycorrhizal colonization. So interestingly, the inoculant with the lowest propagule density had the highest colonization levels. And what we can do with this data, so what we showed was that in the local field soil, which is representative of what could be a future vineyard, there was almost no inoculation potential. It had probably less than 2% colonization here. So what this suggests is that if you're interested in using mycorrhizal products, using any one of these products or even local vineyard soil, adding to this soil seems like it would add mycorrhizal fungi back into the soil that doesn't appear to be there right now. If you're already in an established vineyard where you already have pretty good levels of mycorrhizal colonization, if you wanted to increase that, it seems like these two products could be a good choice. And you could also potentially take local vineyard soil, like from an established vineyard, and move some of that into local field soil. And the way you could do that is you could use little scoops of soil at planting in a certain number of plants. And that typically is a way that people can do these types of soil transplants. So what we found was that these products varied quite a bit in the number of species, the propagule density, and the percent colonization of roots, which had pretty much nothing to do with how many propagules were there to start with. The product with the lowest spore density had the highest inoculation potential, and the local field soil had the lowest mycorrhizal inoculation potential of all. And so this is an example of where, if you were interested in using mycorrhizal fungi, this could be a good place to use inocula. We also found that these inoculant products varied in nutrients. So we did nutrient analysis of all of these products compared to the local background soil and the remnant soil and the vineyard soil. And what I did here was I highlighted the high levels in orange and the low levels in gray. And this is compared to the background field soil. So this local soil that was collected from the WSU campus. And so some of these products have high levels of different nutrients. So it's possible that some of these inoculant products may appear to work not because of the mycorrhizal fungi, but because you might be adding a lot of nitrogen, for example. So there are all sorts of variations in these different products. And so it's important to test them to see if they work in your particular system before applying them on a large scale. So what we're going to be doing this spring, Madeline is my master's student who's going to be growing merlot in live field soil. And she's going to be inoculating each of these pots based on the amount of inocula recommended by each manufacturer. And then she's also going to standardize it based on this mycorrhizal inoculation potential, because we already know that more spores doesn't necessarily mean more colonization. So she's going to do it in these two ways and then see if colonization or plant growth varies in any of these different treatments. So that experiment is coming up this spring. So in summary, in our first experiment, we showed that merlot was more responsive to mycorrhizal inoculation than chardonnay. We had higher shoot growth in the no fertilizer treatment when you add mycorrhizal fungi, and higher root biomass with a combination of mycorrhizal fungi and phosphorus fertilizer. We found that phosphorus fertilizer really reduced mycorrhizal colonization in the roots, but when it was added in combination with mycorrhizal fungi, it did increase root biomass in merlot, which could be beneficial in certain situations. These inoculant products vary widely in propagule number, nutrient concentration, the species of fungi that are there, the number of different species. So what we recommend is, if you're interested in these products, testing them on a small scale first or in a greenhouse experiment to see if they're actually to plants in your soil type, for example. It's also brought up some other questions. So why does Chardonnay have lower growth response to inoculation than Merlot? And these are just the results of this study that we did. It's a short-term study in the greenhouse. Other studies in the greenhouse have shown that Chardonnay does respond to mycorrhizal fungi. And so this experiment used a different species of fungi than what we had. And so Chardonnay appears to respond strongly to that species, but not the mycorrhizal inocula that we used. We also looked through the literature to see if this was a common phenomenon of whether mycorrhizal fungi plus phosphorus fertilizer is associated with increased root biomass. And in several recent studies, that does look to be the case. So adding mycorrhizal fungi in addition to phosphorus fertilizer could be beneficial in terms of root biomass. And in this experiment, we found that these young potted Merlot vines in the greenhouse benefited from inoculation in this particular experiment, but we don't know if these benefits will be observed in the field. And so it's a lot, there's a lot more going on in the field than under controlled greenhouse conditions. So that'll be future directions. So how this is important for local viticulture is that mycorrhizal inoculants may be beneficial to certain cultivars with or without fertilizer applications, but it's important to test that before you apply widely. And the impacts of increased root biomass in the field, maybe that could be important for nutrient acquisition or nutrient retention. And one thing about inoculants is they may require fewer applications than fertilizer because theoretically, once you add them to the soil, and if they associate with the plant, they're going to be propagating themselves as long as nothing kills them. But it also depends on whether they can persist in the field because they're also the resident fungal organisms. And so that's sort of an open question of if you add these to the field, how long do they persist? How do they compete with resident soil organisms? So what we recommend is conducting on-site evaluations of mycorrhizal inoculants before applying them on a broad scale. And you can do this fairly easily, like you could do it in pots, either in a greenhouse or outside, or in a small area of a field, just to test to see if the inoculant seems to work in your situation. So I'd like to thank the graduate students who have contributed to this work. So Casey led the first experiment and also contributed some nice slides to this presentation. And Madeline Luke is my current master's student who is running the second experiment. And I'd like to thank my funding sources, the Washington Grape Washington State Grape and Wine Research Program from the Washington State Wine Commission. The plants were donated to us by Inland Desert Nursery. Fertilizer and some of the inoculant products were provided by Blail Co-op. We've been working with Michelle Moyer, Sarah Del Moro, and Catherine Jones on this project. And we want to thank the facilities staff at WSU Tri-Cities, and special thanks to this group of undergraduates who's helped so much with the project. So thank you for your attention, and I would be happy to take any questions.
Okay, great. Thank you very much, Tanya. There is one already in the Q&A, and then there was a little bit in the chat, but do inoculants need to be applied at planting, or can they be used at any time?
So people use them in both ways. I think they're probably most effective at planting, because a lot of times after field preparation, for example, if the field is tilled, or if it's being planted into a previously agriculturally managed system, those type of management practices can have a negative impact on the resident soil organisms. And usually the only ones that can survive that type of disturbance tend to be more of the weedy, stress tolerant species. So applying inoculants at the time of planting, if it's been shown to help improve your particular type of plant growth, I think that would be a good strategy. But they can also be used anytime. I've seen people apply them later as well. And so people are using them more and more as sort of like a biofertilizer.
There was one question about, does ortho or polyphosphorus make a difference?
Oh, that I don't know. That's a good question. I'm going to write that down. I'm not sure. Thank you.
Okay. And then can you maybe touch on the difference between spores and propagules? Because obviously spore count is a little different than the propagule count. So how does that vary?
Yeah, so on the product packages, sometimes they talk about spore count, and sometimes they talk about propagule count. And propagules are spores and hyphae and root fragments, anything that can colonize, that hosts mycorrhizal fungi and can colonize a plant. And so one of the things we're going to be doing also, let me see if I can go back here. I showed that some of these things are grayed out. Yeah, so we're going to be doing spore extraction. So we're going to be getting the actual spore numbers from each of these products. We just haven't done that yet. This is what's on the package.
Okay, great. Would you recommend applying them as a dip at planting in commercial vineyards?
Oh, that's a good idea. The only thing is that these mycorrhizal fungi are aerobic, so you wouldn't want them sitting in water for a very long time. Like you wouldn't want to make a dip and let it sit there for a week and then add that to your plants, because it can get anaerobic and the fungi can start to die and rot. So a lot of times it's used as a like granular application. But if you're using it with water, just don't use it. Don't let it sit too long, I would say.
Okay. Does colonization increase if the inoculant is made during the warmer time of the season, you know, spring, early summer versus maybe fall when it would be colder? Does temperature have a difference, soil temperature?
On mycorrhizal colonization levels?
Yes.
Okay, so that's a good question. So these products are generally made in greenhouses. So the only way, because mycorrhizal fungi are obligate symbionts, you can't culture them on a plate, for example. So you have to grow them with plants in a greenhouse. So most of the commercial inocula are grown, are the fungi that are easiest to grow and bulk up and under greenhouse scenarios. In the field, the level of colonization has a lot to do with photosynthesis and photosynthetic rate over the growing season. So in areas of, peak summer where there's a lot of photosynthesis, that's a lot more carbon going down into the roots to support the fungi. Whereas later in the season, especially for annual plants or plants that drop their leaves in the fall, as the plants slowly sort of slow down photosynthesis, that can trigger the fungi to sporulate. So what we do is when we look at mycorrhizal colonization levels in roots, you can look at the different structures and it can sort of give you a clue as to what's happening. I'll see if I can find a picture here really quick. Yeah, here we go. So in this root sample, we see a lot of these little cloudy areas, and these are called arbuscules, which is the site of nutrient exchange. So at this sampling, there's a lot of nutrient exchange that looks like it's happening. These are hyphae that go out into the soil. So if you see a lot of hyphae, that could indicate that there's a lot of sort of foraging for nutrients. Later in the season, you start getting these structures. There aren't any here, but they're called vesicles, and they look like little balloons. And that's when the fungi start to get sort of stressed out. The plants are shutting down, and so they make these vesicles and then spores that then rest in the soil or roots until the next year. So it does definitely vary over the growing season. But if they're in the soil, they come back in they are still associating with the plants. It's the colonization level tends to vary with photosynthesis.
Okay, another one. Do you have any experience using biochar in addition to mycorrhizals?
Yes, one of my other graduate students did this he used a native, it was in a mining restoration experiment. So he was looking to see if biochar with and without mycorrhizal fungi improved plant growth in these toxic mine soils. And what he found was that there wasn't really any sort of synergistic effect. So the biochar didn't enhance mycorrhizal inoculation or anything like that. But the fungi were able, once they were added back into even this toxic mine soil, they were able to form these associations with roots. So it was sort of similar here, where we had the contaminated mine soil, and the plants grown in that had no colonization. If you added mycorrhizal fungi with or without biochar, then they got mycorrhizal colonization. So if your area or your soil is deficient in fungi, if you add it back in, even under, it seems like they can still colonize the plants and help them get established in the soil.
Okay, we've got another one. Is there any research to identify what AM fungi species are most associated with vitis, and are ectomycorrhizal species associated with grapes, vitis?
So the best way to identify the fungal species that are associating with roots would be through molecular sequencing. You can do spore extractions, but it's really hard to identify these fungi to species using the spores because they're all little brown, orange, yellow spheres. So it's hard to identify them to species. And then also, just because something's in the soil doesn't mean it's in the roots. And so people tend to use molecular sequencing to identify the fungal species that are in those roots. And mycorrhizal and the grapevines associate with arbuscular mycorrhizal fungi. Ectomycorrhizal fungi tend to associate more with trees.
Okay, great. Let's see.
But that's a good question when you're looking for inocula, because some inocula is ectomycorrhizal inocula and some is endo, which is the arbuscular mycorrhizal fungi, or some's a mix. So that's a good thing to keep an eye out for if you're specifically interested in using inoculants with grapes.
And are there added benefits to using these products in organic versus conventional vineyards? Do you see a difference?
I would say, it depends on soil fertility levels, because so in organic farming systems, they tend to use fertilizers that are like more slow releasing, like compost, for example, whereas in conventional growing situations, people tend to use commercial fertilizer where there's a lot of nitrogen and phosphorus going in at once. And so if you're looking to maximize the benefits of mycorrhizal fungi, it would be in a more low nutrient soil that's not getting these pulses of large quantities of nutrients. But the fungi are resilient too. So, once those pulses are done, if they're still in the soil, they can come back. But they're going to be most beneficial to plant growth when there's not a lot of extra nutrients in the soil.
Okay, and then could microbloom or the Mycos Gold be applied as a root dip? Would they stay in suspension long enough? I think you kind of touched on that. But
yeah, so microbloom, sort of has the consistency of kitty litter. So, but that's a lot of the filler. So the fungal spores maybe would float or stay in suspension, but the filler would sort of sink to the bottom. So most of the time, actually every time that I've worked with mycorrhizal fungi, we use it as a granular formulation. So I don't know as much about using it as a root dip or not. But again, as long as they're not sitting in water for too long, and yeah, I suppose you'd want to keep it mixed up so they don't just all settle out.
Okay, let me see if I've got any more. I think we've got them all. Yeah, great. And we're right at 1:30. So perfect timing, very informative, Tanya and Dr. So as I said, we look forward to her wrapping up the last half of this project, and she will be putting a report together. We will share that, and then there's probably will be enough interest just for that portion of the research. We can do another webinar when all that's wrapped up. So thanks. And we've recorded this, so you will get a link to the recording if you want to watch it again. And for those, and it will be posted up on the Wine Commission's website in the near future. So with that, get out there and start growing those vines. Thank you.
All right, thank you so much for having me, and for those who joined today.
Okay, adios.